Right. Good day, grade 12s. Welcome to class, to a new session of which today we will be looking at a subtopic called DNA replication. Now, I just want to make a mention that DNA replication is a continuation of the module or the topic that we've been looking at, which is DNA code of life. Okay, so it's also very important that we try to put together the ideas, or rather the knowledge that we have acquired this far about the nucleic acids, that is the DNA and RNA, if we can ever stand a chance to understand this topic or this subtopic called the DNA replication. Okay, now what is DNA replication? These are the questions that we'll be answering. What is DNA replication? And why does DNA replication take place? And where? So these are the questions that we'll be answering here. The question of what is DNA replication. 2, we will talk about where. When you talk about where, we simply talk about where does this process take place. Then why. You see, why does DNA replication take place? Now, in simple terms, what? DNA replication is the process by which DNA make a copy, make an identical copy, make identical copy of itself. So DNA makes an identical copy of itself. That is DNA replication. So we've answered that question of saying what. And where does DNA replication, remember about the location of DNA. DNA's location, remember when we were introducing the concept DNA or nucleic acid, we actually paid much greater attention in identifying where in the cell DNA is located. And we identified a couple of, we identified 3 main organelles where DNA is located. And we talked about the nucleus, as well as the mitochondrion and the chloroplast. But we are not concerned about the last two, meaning the mitochondrion and the chloroplast. Why? Because that is that extra DNA that is found there. But the main DNA that we're talking about here, where DNA replication takes place, is none other than the nucleus. So the DNA located in the nucleus. So that is where DNA replication takes place. Remember, DNA replication takes place in the nucleus. It takes place in the nucleus, right? Now, before we look at the question of why, I would want us also to look at the question of when. When does DNA replication take place? When is simply talking about the phase or the stage at which this process takes place, and this occurs during a phase called the interphase. So it takes place during what? Interphase. Right. Good. Now here's the last portion that is remaining. Why does DNA has to make an identical copy of itself? There are two reasons why DNA has to make an identical copy of itself. First, or one, is that DNA makes an identical copy of itself so that it is doubled. So DNA replication doubles the DNA. It doubles the DNA. That's number one reason. So to double, can you think of um, a copy of a paper that you might have for an activity? So that copy of a single paper, if you want to make copies, what would you do? I guess you would take it to a photocopier, or you'd put it on a photocopier machine, isn't it? So the photocopier machine is going to make another copy that is going to be identical of the original copy. So what has happened to that copy? The copy has been duplicated, or meaning that you make another copy that is identical. So DNA replication doubles the DNA. Now we must not forget that the DNA is the genetic material, isn't it? Because DNA is passed from one generation to another. So before this DNA is actually passed from one generation to another, it has to be doubled. That's why DNA replication actually has to take place. Right, that's number one. Number two, now here's the DNA that has been doubled. So what? So the DNA that has been doubled, it is doubled so that it can be shared, so that it can be shared between resulting cells, between resulting daughter cells. Now remember, this process actually is also a link between mitosis, which is cell division, as well as the process of meiosis, where gametes are formed. Right. So therefore, it's very important that DNA is actually doubled. Why is it doubled? So that it can be shared between the resulting daughter cells. Okay. Right. Now, when does it take place? Remember, it takes place during interphase. Now, the process of interphase is a process that occurs way before the process of before mitosis. Remember, I talked about mitosis as cell division. Before mitosis, before the process of meiosis, which is gamete formation. Okay. So now we know why this process is very important. So the significance of the process of DNA replication, it hangs around what we call it hangs around the why. So if in the examination you are asked to say, can you state the significance or explain two significance of the process of DNA replication? Remember, in that case, you'll be answering the question why. Okay, good. Now, here is the important segment of this process. So it is very important that when you are describing the process of DNA replication, you need to remember the stages. You need to remember the what? The stages. Now, these stages actually they describe the process, the whole process, how it occurs, isn't it? Now, Let's begin here. If you remember very well in what we learned that DNA in its originality, it is a double helix. It is a double helix, meaning that it is twisted like a rope, isn't it? There you go. This is my DNA. So this DNA, it has to unwind before anything else happens. The DNA, it needs to untangle itself because it is twisted like a rope. So it needs to unwind. So the first point is that the DNA unwinds. So when it unwinds, it means it becomes a double-stranded, where it is consisting of two strands. I've got strand 1 there and I have strand number 2. So I've got 2 strands there, strand 1 and strand 2. These are the 2 strands in a double-stranded structure, of which these 2 strands, they are joined by the weak hydrogen bond. This is the hydrogen what? Bond. The hydrogen bond. Okay. So now in this case, because now the DNA is actually unwinds, then now it actually becomes a double-stranded. So the double-stranded DNA, it actually unzip. Can you think about the zip of your jacket? The zip of your jacket when you are cold. Oftenly, you close the zip, isn't it? So that the cold does not get into your body. So the zip actually closes. Now, if you feel hot, what happens? You bring down your zip. So the teeth of the zip, they unlock. So I'm just trying to relate to this, to relate the zip situation into the DNA. So the DNA, which is actually joined by the DNA strands, which are joined by the weak hydrogen bond. When the weak hydrogen actually bonds breaks, we say this allows the DNA to unzip. You get what I'm saying then? So the first thing here, we talked about unwinding. Then the DNA actually is weak hydrogen bond breaks. So this allows the 2 strands to do what? To unzip. So that's very important. So what happens when it unzip? The DNA actually forms 2 separate strands. So you're going to have how many strands? We've got strand number 1 there and we've got strand number 2. This strand, the first one, is the one that we're going to have as strand number 1. So both of these strands, they are going to act as templates. They are going to serve as what? As templates. So now, one would be asking that, say, what is a template now? Now, simple. A template is a DNA strand, like the ones that we have. Remember, I'm saying that we've got 2 templates here. So these 2 DNA strands are templates. So a template is a DNA strand that is used to form another DNA strand. Now, because now these have separated them, there are 3 nucleotides that we are going to talk about. There is within the nucleus itself, in the nucleoplasm. You have actually the 3 nucleotides. You got the T, you got that thymine, you got adenine, you got cytosine, you got guanine, adenine, thymine, you got guanine, you got there are many within the nucleoplasm. Now, you know that the base pairs in a DNA, A bonds with T, or vice versa. Then G bonds with C. So in this case, the 3 nucleotides, they are going to line up there, whereby A is going to invite T, T is going to invite A, G is going to invite C. and C invites G. It goes on and on like that, where the base pairs, they actually link up as the way they are supposed to bond, like I've indicated. Right. So this is going to continue. I guess you can actually continue there to write the base pairs that are going to bond with these others, isn't it? Very important. Now, these at the end of the day, they are going to form a new DNA molecule. So now, this DNA molecule actually, now with the free nucleotides having bonded together with the hydrogen bond, now it ends up forming a new DNA molecule like I've indicated. So that is what happens on this one there. Now we've got one new DNA molecule. Now, in strand number 2, the same thing that has happened to the first strand, it also happens to this one, because now the DNA strand, which is a template, is got to free bases. So the free nucleotides also within the nucleoplasm, they are going to be invited, right? Whereby we are going to have a T inviting A inviting T, C inviting G, and G inviting C. It goes on and on and on according to the way they are complementary to. So these new strands that have formed here, these new strands, we call them templates. They are called what? Templates. So you've got the template here, which has formed the DNA, the complete DNA molecule, and we have another template there, which has formed a new DNA molecule. Now, I guess you can actually continue there to form, you know what A bonds with, T bonds with who, G bonds with, and C bonds with whom. So eventually, we are going to have another DNA molecule. So how many DNA molecules do we have? We have 2 DNA molecules. Now, if you watch carefully, or if you observe carefully here, you will discover that you've got A there, you've got T there, you've got A there, you've got T there. T there, A there, T there, A there. Can you see that? So in this case, you can see that these pairs are identical. Thus, these 2 DNA molecules, they are going to be identical. They are going to be identical. So these 2 molecules now, they are going to be used in the corresponding processes which we have mentioned earlier. I spoke about mitosis and as well as meiosis. So these 2 processes, they are going to use these DNA molecules in order to form new cells, which we call the daughter cells. So during mitosis, the daughter cells are going to be identical, meaning that they are going to be similar. Why are they similar? Is because, remember, they are sharing identical DNA molecules. So that is how you would describe the process of DNA replication. Okay, so now what you can do is you have to go to a past exam paper and try to get a question on DNA replication, and try to see if at all you can answer questions for yourself. Right? Thank you.